So this is the first of the videos which are going to be on enzymes, and enzymes are part of session 2, but they're part 2 of session 2. And once again, we're going to look at the same statement of inquiry, which is all about the use of scientific models. So models, remember, are representation, simplification of an idea. So here we're going to look at the model of an enzyme, and we're going to look at the structure of an enzyme modeled by using a term called the active site of the enzyme. We're also going to appreciate how that model and the active site allows us to understand how enzymes function. And we need to think in particular about this idea here, which is referred to as the lock and key hypothesis. So the lock and key hypothesis is an analogy, which is another kind of model about how enzymes can do their job, how they can function. For GCSE, this is the syllabus statement that we need to know. So let's unpack it just a little bit here. So we need to think about the role or the job of an enzyme. So we need to think about what is an enzyme and what is its job. And its job is to act as a biological catalyst. Biological means it's found inside a cell or a living thing. And a catalyst is something which speeds up a chemical reaction but remains unchanged at the end. So enzymes are biological catalysts. They speed up reactions which are found within our cells. And the reactions within our cells allow us to do our metabolism. And the metabolism are the life processes, which is what we already know from another model, Mrs. H-Gren. So Mrs. H-Gren sums up our whole metabolic processes, our whole metabolic reactions that take place in our cells. So all of the work that we're going to do on Mrs. H-Gren is related somehow to enzymes. So enzymes are biological catalysts. They speed up the rate of a chemical reaction. They remain unchanged at the end, and they're essential to life. They're essential to the metabolic processes that underpin life. These are the questions that we need to be thinking about now, and we come back to at the end of this unit and just check in. But the factual question is, how do enzymes work? And we're going to look at how models allow us to understand this. The conceptual question is what is a paradigm shift in the sense of an enzyme? And a paradigm shift is a change of approach or a change in a way of thinking. So how have we changed our ways of thinking about enzymes in the scientific community? And the last question, very philosophical, to what extent can we observe chemical processes in a cell accurately? So it's thinking about how we can see inside cells using technology to allow us to understand the life processes. So the first job for you, I want you to pause the video in a moment. I want you to then pick out your textbook, read pages 6 to 7. In our Google Classroom, I've posted a 1-minute enzyme animation, which I'd like you to take a look at. And then I want you to watch the second enzyme, which is obviously spelt wrongly there, pardon me, to watch a 4-minute video, which is the Fuse Schools video. Once you've done all of these 3 things, I want you to write down questions that are occurring to you, thoughts, ideas that I want you to bring back into our Google Meet. I don't yet want you to make notes. So I want you to pause the video, read the textbook, watch these 2 links which I've posted in the Google Classroom, write down your thoughts, and then once you've done that, you can carry on with the next part of this video, and we'll bring those ideas and those thoughts that you've got into the plenary section that we have of this lesson. I'm hoping that you've got some good questions, and I'm hoping that you've learned a little bit from looking at those resources. So remember that what we're looking at here is the enzyme as a biological catalyst. So we need to think about what they are and what they do. Obviously, you can read this for yourself, but let's just pause for each one. So they speed up chemical reactions of metabolism. They speed up chemical reactions which are happening inside our cells. We need to also think about the fact that enzymes, even though they are involved in a chemical reaction, they remain unchanged at the end so that they can be used over and over again. So you only actually need a small quantity of enzymes to do many chemical reactions. What we also need to think about are all enzymes are made of protein. So one of the reasons that we need protein in our diet is for growth, for repair, and also for making enzymes. And the other thing to bear in mind are enzymes are specific, and we're going to come to look at how that specificity works in the following couple of slides. So what are enzymes doing? Well, they control all the chemical processes of life, and I've just listed 4 for you to think about, which are big ones in terms of the IGCSE course. So respiration is a chemical reaction that allows your cells to produce ATP. That's energy, that's the energy currency of the cell. We're going to look at respiration in detail in year 10, but it's enzymes that are allowing you to do that particular chemical reaction. In plants, the reaction of photosynthesis is also dependent on enzymes, which again we look at in year 10. Then we've got this idea of digestion, which we've sort of touched on when we look at balanced diet and we dig back into later on. But digestion is breaking down large molecules into small molecules that can be absorbed into our bloodstream. And digestion is dependent on enzymes. When we get to year 11, we're going to look at DNA replication and protein synthesis. And once again, these chemical reactions are powered by or rely on enzymes. So let's start thinking about models in terms of the enzyme. So the green blob here, which looks a bit like Pac-Man, this is meant to represent an enzyme. And enzymes are globular. They're globular proteins which are found inside the cells, but they've got a very, very specific shape. And this specific shape, as part of their structure, is referred to as the active site of the enzyme. Now, all enzymes, each individual enzyme will have a different shape for a different active site. And in each case, the active site is specific to a particular reactant, or in this case, when we talk about enzymes, we talk about substrates. So the thing that is going to be worked on in the chemical reaction, the thing that's going to change the reactant of the chemical reaction, must have a shape which is complementary to the shape of the active site. So I want you to think about this idea of the lock and the key hypothesis. So if you've got the right key, you can open up the lock and open up the door. If you've got the wrong key, you can't open up the door because the lock doesn't fit the key. And it's the same idea here. So what we've got is the right substrate being held by the enzyme, because the active site of the enzyme and the shape of the substrate are complementary. And this is called an enzyme substrate complex. And because of this interaction of the enzyme and the substrate, that allows what we call a lowering of the activation energy, and that allows a reaction to take place. And here we're producing a product. So here's an enzyme. Here it's exactly the same as here. It can be used over and over again, but we've got the substrate, the reactant, being converted into a product. And this particular example uses an example which is a digestion example. So we've got a large molecule here, and it's being broken down into 2 smaller product molecules, which are just here. So the lock and key hypothesis means that the active site of the enzyme is complementary to the shape of the substrate. That allows us to form an enzyme substrate complex, and the enzyme substrate complex lowers the activation energy that allows the enzyme then to produce 2 products and remains unchanged at the end. So continuing and adding another layer of detail, we need to think about if we want the substrate to react with the enzyme, the first thing we've got to do is get them to meet each other, and we've got to get them to attach to each other like so. So the enzyme needs to collide with the substrate in order for the reaction to take place. And they need to collide with enough energy so that we can form this complex and lower the activation energy. So we talk about the kinetic energy that is required for the two to collide. And kinetic energy is movement energy. So we want to think about, and we'll come back to this later on, the idea of temperature. If we've got very cool temperature, then the substrate and the enzyme are going to be moving slowly. If we warm the temperature up, we give these more energy, and if they've got more energy, they're more likely to collide and the reaction is more likely to proceed. So there needs to be, for a reaction to take place, a collision between the enzyme and the substrate, and that collision is going to be reliant on the kinetic energy of the system. Here we've got the same sort of idea, but again, we've got a little bit of extra detail. We've got the enzyme on the substrate, the complex. Now, when we have this complex being formed, it turns into what is referred to as a transitional state. So the substrate collides with the enzyme, get an enzyme substrate complex, and the interaction, the specific chemical interaction between the substrate and the enzyme allows what's called a transitional state. And that transitional state lowers the activation energy to allow us to produce the product. Now, what I'd like you to do is in your booklets, I want you to read the first couple of pages, but you will find a table which looks like this. And what I need you to do is to look at each one of these terms, and I want you to match these terms with a definition. Now, to help you, I've posted in the Google Classroom a set of definitions. I've muddled them up, so you need to look at that extra sheet and match the enzyme with the definition on the help sheet, which is in the Google Classroom. Once you've done that, what I'd like you to do is to make some extra notes. So I want you to go back into Google Classroom. I want you to watch the 8-minute video, which is named Science Biology 9 to 1. 9 to 1 refers to your syllabus. Then you've got a 4-minute video, which is named Enzymes GCSE, also 9 to 1. You might want to re-watch this 4-minute video that you looked at earlier on. I want you to make notes on the video. I want you also to be carefully using your textbook. And again, have a look at the questions that you've got, bring them into the Google Meet session. And by doing this particular note taking, I'm sure that you'll have even more questions that you can bring next time.